T lymphocyte T cell activation and differentiation. The adaptive immune response has very important attributes such as specificity and memory response. The T cells and B cells are the major army of adaptive immunity. The T cell and B cell recognize specific antigen and get activated, in which they undergo clonal expansion and proliferation to form clones of cells which differentiate into effector and memory cells, where effector cells specifically help to neutralize the antigen, while memory cells help immune system to memorize the particular antigen and helps to respond at a faster rate in subsequent exposure with the antigen. In the present module, we will try to understand how T cells specifically respond to initial binding of antigen, how it differentiates into different types and performs its specific action. Key events in T cell activation. Antigen recognition by T cells. A single positive CD4 plus or CD8 plus T cells develop in rhythm in timers and enter the circulation as resting cells in the G0 stage of the cell cycle. The T cells which have not encountered antigen are known as naive T cells. The naive T cells have small cytoplasm and little transcriptional activity. The antigen recognition by T cells is majorly done by T cell receptor TCR complex along with the help by additional molecules such as co-receptor CD4 in case of T helper cells and CD8 in case of cytotoxic T cells and addition molecules. T cell receptor complex. T cell receptor is present on the surface of T cells. It has highly variable alpha and beta heterodimeric chain, which is chiefly responsible for recognition of processed peptide present on MHC molecules by antigen presenting cells. But TCR is not complete with alpha beta chain. It requires the presence of other chain to express on the surface of T cells. The other invariant chains include CD3 gamma, CD3 delta, and CD3 epsilon chain. Together form CD3 complex. And two disulfide linked homodimeric zeta chains. The zeta chains have very short extracellular domain and long intracellular domain. The heterodimeric alpha beta chain along with invariant chains CD3 complex and zeta chains constitute T cell receptor complex. The alpha beta chain interact with CD3 delta chain and CD3 epsilon chain, CD3 gamma and CD3 epsilon, and homodimeric zeta chains via reciprocal charge interaction. The intracellular domain of CD3 complex and zeta chains have immunoreceptor tyrosine based activator motifs or immunoreceptor tyrosine based activator motifs. Immunoreceptor tyrosine based activation motifs. Each ITAM has two tyrosine residues which are sites for phosphorylation by protein kinase. The two tyrosine residues in each ITAM are separated by 9 to 12 amino acids and presented as like this. So where Y is tyrosine, L is leucine, I is isoleucine, and X represent any amino acid. Once phosphorylated, the two tyrosine residues help in recruitment of signaling proteins that contain two tandem SH2 domains. The alpha beta chains recognize specific antigen while invariant chains help in downstream signaling events. TCR recognition CD3 alpha beta gamma delta epsilon. Epsilon delta alpha beta gamma epsilon. CD3 TCR recognition ITAMs signaling. So T cell receptor complex. T cell receptor with alpha and beta heterodimeric chain with short cytoplasmic tail, CD3 complex, CD3 gamma, CD3 delta, and CD3 epsilon chain, and two disulfide linked homodimeric zeta chains with long cytoplasmic tail. Then co-receptor CD4 or CD8. The alpha beta chains of TCR complex is helped by co-receptor CD4 or CD8 in performing their function. The co-receptor CD4 or CD8 help in stabilization of the interaction between alpha beta chains and processed peptides on MHC molecules via their binding to invariant sites on MHC molecules. Further, they also help in signal transduction after antigen binding, as they are associated with non-receptor tyrosine kinase of SRC family. Besides co-receptor, there are additional accessory molecules on T cells which interact with additional molecules on antigen-presenting cells with further stimulate antigen recognition and downstream signaling events. Together, they play important role in T cell activation. Then events after antigen recognition. Initiation events. T cell recognizes processed specific antigen presented by MHC molecules on the surface of antigen presenting cells via the alpha beta chains of TCR complex. The binding event also involves CD4 or CD8 co-receptor on T cells as they bind to invariant regions of MHC molecule. During this recognition process, the following changes happen. Non-receptor protein tyrosine kinase belongs to SRC family such as I, CK and FIN present in cytoplasm or associated with cytoplasmic tails of the co-receptors. CD4 and CD8 get stimulated. Also CD45 common leukocyte antigen protein phosphatase helps in dephosphorylation of C-terminal tyrosine residues of PTKs such as ICK and FIN and make them active. The recognition event also led to conformational change in cytoplasmic tail of CD3 complex chains and zeta chains, which in turn exposes its ITEM, two tyrosines flanking a series of amino acids with particular spacing. The initial binding events lead to activation of downstream signaling events. Antigen presenting cell MHC2 molecule, alpha beta CD4 extracellular site, cytoplasmic site, TCR complex, cytoplasmic site, downstream signaling events. T cell activation. Antigen recognition as extracellular site and downstream signaling events on cytoplasmic site. Antigen recognition is helped by T cell receptor complex, co-receptor CD4 and CD8, CD45 and other molecules which are not shown. So downstream signaling events are mediated by cytoplasmic tail of CD3 complex, zeta chains, co-receptor CD4 and CD8, CD45 molecule. CD8 will be there. So the PTKs phosphorylate tyrosine sites on each ITAM present on cytoplasmic regions in CD3 complex and zeta chains of TCR complex. The two phosphorylated tyrosine sites on each ITAM serve as docking sites for signaling proteins with two SH2 domains such as protein tyrosine kinase, SYK kinase family member, PTK such as phosphopoietin ZAP70, zeta associated protein of 70 kilodalton. The ZAP70 gets phosphorylated and activated by ICK. The ZAP70 then catalyzes the phosphorylation of number of membrane associated and cytoplasmic adapter proteins, which leads to recruitment of several intracellular signal transduction pathways, which leads to T cell activation. Extracellular side, cytoplasmic side, LCK, ZAP70, antigen presenting cell, MHC2 molecule, antigen recognition, conformational change in cytoplasmic tail of CD3 complex and zeta genes, recruitment of the co-receptor CD4 associated kinase LCK, phosphorylation of ITAMs in CD3 complex and zeta genes, recruitment of ZAP70, phosphorylation and activation of ZAP70 by LCK. So initial signaling events after antigen recognition, it leads to ZAP70 activation. The ZAP70 catalyzes the activation of several proteins, which in turn lead to recruitment of several intracellular signal transduction pathways and initiate downstream events. ZAP70, zeta associated protein of 70 kilodalton. The most important targets of ZAP70 are the transmembrane adapter protein linker of activated T cells, LAT, with long cytoplasmic tail, and cytosolic adapter protein SRC homologue to SH2 domain containing glucoside phosphoprotein of 72 kilodalton, SLP76. The LAT and SLP76 can also be linked via adapter protein GATs. These molecules are responsible of activation of multiple signaling pathways. The importance of these molecules can be understood by the complete absence of T cell activation signaling events in mice double deficient for LAT and SLP76. The LAT and SLP76 can also be linked via adapter protein GATs. These molecules are responsible for activation of multiple signaling pathways. The importance of these molecules can be understood by the complete absence of T cell activation signaling events in mice double deficient for LAT and SLP76. On TCR engagement and ZAP70 activation, LAT gets phosphorylated on its 9 tyrosine residues. The phosphorylated tyrosine on LAT bind to various effector molecules such as C-terminal SH2 domain of phosphorylipase C-gamma-1, P85 subunit of phosphoinositide 3 kinase, PA3K, adaptase growth factor receptor bound protein, GRB2 related adapter downstream of SHC GATs. So SLP76 also gets phosphorylated and interacts with a large number of effector molecules such as SH2 domain of VAV1, NCK and IL2 induced tyrosine kinase ITK, motifs and binds to GADs and PLC gamma 1. The LAT and SLP76 along with effector molecules serve to stabilize proximal multimolecular signaling complex that help to mediate the downstream signal transduction. This is LAT and SLP76 along with effector molecules. The LAT and SLP76 is activated by ZAP70. Activated LAT leads to activation of membrane-associated phospholipase C-gamma, PLC-gamma, adapter protein GATs and other molecules. Adapter PLC-Y, LAT phosphorylation, GATs, SLP76 and ITK. While activated SLP76 interact with IL2 induced tyrosine kinase ITK, GATs and phospholipase C-gamma, PLC-gamma. Then downstream signaling events. One of the signaling protein, phospholipase gamma, PLC gamma 1 carries the signal forward. PLC gamma 1 gets activated via ITK, a member of TEC family of the cytoplasmic protein kinase. ITK is recruited and activated via several ways. The ITK is recruited to the membrane by interaction with SLP76 via their SH2 and SH3 domain and gets phosphorylated by LAT and SLP76 scaffold. The ITK is also recruited via their pH domain, which interact with PIP3 on the inner face of their membrane, which is generated by the activation of phosphoinositide 3 kinase PI3K by LAT and additional accessory molecules described later in detail section co-stimulation and gets phosphorylated by ICK. The activated ITK phosphorylates PLC gamma 1 and make it active. So activated PLC gamma 1 hydrolyzes phospholipid component of the membrane phosphatidylinositol 4,5 biphosphate PI 4,5P2 to generate the second messenger inositol 1,4,5 triphosphate IP3 and diacylglycerol DAG. The second messenger activates several distinct signaling pathways which results in expression of diverse set of genes required for T-cell activation. The three important signaling events are as follows. DAG-mediated signaling pathway. Diacylglycerol generated by PLC gamma 1 diffuses in the plasma membrane and activates a variety of protein which further leads to activation of intracellular major pathways involving RAS and protein kinase C, PKC. So DAG activates RAS GRP guanine nucleotide exchange factor that activates small GTPase RAS. RAS a small GTP binding protein and is required for the initiation of mitogen associated protein kinase MAP kinase signaling pathways. RAS GRP promotes exchange of GDP for GTP in RAS and made it active. Once activated, RAS activates the serine threonate kinase RAF1, mitogen associated protein kinase MAP kinase, which then activate MAP kinase 1, MAP K kinase, MAP kinase, which further phosphorylates a tyrosine and a threonine residues on MAP kinase ERK1 and ERK2. Extracellular signal related kinase. ERK kinase results in the phosphorylation and activation of transcription factor ELK1, which is necessary for the expression of FOS gene. FOS protein gets dimerized with another protein Jan to yield activated heterodimeric protein 1 AP1, essential transcriptional factor for T cell activation. Activated PLC gamma P145P2 DAG activation IP3, then RAS GRP, inactivated RAS, active RAS, MAP kinase, MAP kinase. Then increased expression of FOS gene activation of ELK transcription factor, MAP kinase, cell activation, T cell activation, AP1, GUN, then mRNA, ribosomes. Force. Activated PLC gamma 1 lead to hydrolysis of phospholipid component of membrane 1, 4,5-P2, which generates diacylglycerol and IP3. Diacylglycerol diffuses in the membrane and tend to activation of FAS followed by MAP signaling pathways, which in turn contribute to T cell activation. TAG also activates an isoform of protein kinase C, PKC contains lipid binding domain specific for DAG, gets activated in the presence of DAG. PKC theta regulates NK kappa B NFK beta activation. Generally, in resting cells, NFKB is found in cytoplasm in association with its inhibitor of kappa B, IKB, which prevents moving of NF kappa B into nucleus. PNC eta causes the release of inhibitory molecule, allowing NFKB to translocate into the nucleus, where it promotes the expression of genes involved in T cell activation and survival. Nucleus upregulation of genes for T cell activation and survival. NFK beta, IKB, PKC, theta, DAG, degraded IKB, NFKB. Diacylglycerol also activates NFK beta signaling pathways. DAG activates protein kinase C, which helps in degradation of NFK beta inhibitor IKB. Activated NFKB moves to the nucleus and upregulates genes for T cell activation and survival. Then IP3 mediated signaling pathways. The IP3 generated by TCR activated PLC gamma 1 activity diffuses into the cytosol and binds to IP3 receptors on the endoplasmic reticulum membrane. It causes a rapid release of calcium from the endoplasmic reticulum into the cytoplasm and also opens CA channels in the cell membrane, resulting influx of calcium in the cytoplasm. Activated PLCY1 P145P2 hydrolysis DAG DP3 release of Ca2 plus lumen of endoplasmic reticulum. The IP3 generated by TCR activated PLC gamma 1 activity causes a rapid release of calcium from the endoplasmic reticulum into the cytoplasm. The increased cytosolic Ca results in the activation of a family of transcriptional factors called NF80, nuclear factor of activated T cells. In resting cells, NF80 remains in the phosphorylated form in the cytosol. The cytosolic Ca binds to calmodulin and causes the activation of calcineurin protein phosphatases, which dephosphorylates members of the nuclear factor of activated T cells, NF80 family, which translocates into the nucleus. In the nucleus, NF80 can form complexes with other transcriptional factors, resulting in a differential gene expression patterns for T cell activation, such as Th1, Th2, and T anergic or Treg. Increased Ca2 plus binds to calmodulin, active calcineurin. Inactive calcineurin turns into active calcineurin. Dephosphorylation, inactive NF80, active NF80, differential gene expression, TFS. Activation of NF80, nuclear factor of activated T cells via increased calcium. The increased calcium binds to calmodulin, which in turn activates calcineurin. Calcineurin activates NF80, which can form complexes with other transcriptional factors, resulting in differential gene expression patterns for T cell activation, such as Th1, Th2, T anergic or Treg. The importance of NF80 signaling pathway in T cell activation can be illustrated by the effects of immunosuppressant cyclosporin A and tacrolimus. Both inhibit calcineurin, which in turn inhibits NF80 activation, which in turn prevents T cell activation. The importance of DAG and IP3 in T cell activation can be understood by the observation that T cells activation events can be mimicked by forbol myristate acetate, an analog of DAG, and inomycin, a drug that mimics action of IP3 without involvement of TCR complex interaction with peptide MHC complex. Then co-stimulation, additional requirement for T cell activation. The T cell activation is initiated by engagement of TCR with processed antigen on MHC molecules on antigen presenting cells, which are popularly known as signal 1. In 1987, Helen Quill and Ron Schwitz found that TCR peptide MHC interaction is not sufficient for full T cell activation. The other stimulatory signal besides TCR MHC interaction signal is required for complete T cell activation. The co-stimulatory signal is probably known as signal 2. The other co-stimulatory signal is provided by the interaction between auxiliary molecules such as CD28, ICOS, etc. on the T cell and co-stimulatory ligands molecules such as B7 family members on the APC. The one of the most characterized co-stimulatory interaction is between CD28 and B7 family members who together deliver positive signals to the cell which promote T cell proliferation, cytokine production, cell survival and cell metabolism. So after engagement of CD28 by B7 molecule, the tyrosine residues on the cytoplasmic tail of CD28 get phosphorylated by ICK. The activated tyrosine residues on CD28 activate phosphoinositide 3 kinase PI3K, which generate a PIP3, which has two important effects. It helps in recruitment of ITK via its pH domain to this membrane where it gets phosphorylated by ICK and LAT SLP76 complex. Activated ICK can then phosphorylate and activate PLC gamma 1. Activated PLC gamma 1 helps to activate distinct signaling pathways via secondary messengers DAG and IP3 as described above. It helps in the recruitment of serine threonine kinase AKT, also known as protein kinase B, to the membrane via AKT pH domain. AKT gets phosphorylated and can activate a variety of downstream proteins. AKT signaling helps to promote cell survival by inhibiting the cell death pathway and promote cell metabolism by increasing the utilization of glucose. The signal 1 and signal 2 acts in quantitative as well as qualitative manner to fully activate T cells. Without co-stimulation, T cell become non-responsiveness. If T cell receives a signal 1 in absence of co-stimulatory signal, then T cell comes in state of non-responsiveness called anergy. In experiment, when T cells are incubated with glutaraldehyde fixed APCs, which do not express B7 family members, there is minimum proliferation of T cells resulting in anergic T cells. So anergy can also be induced in T cells when incubated with normal APCs which express processed peptides on MHC molecules as well as B7 in the presence of Fab portion of anti-CD28 which blocks the interaction of CD28 with B7. Once anergic, T cells become unresponsive to normal APCs which can deliver both the signals required for T cell activation. Therefore, interaction of TCR with anergic peptide MHC complex and engagement of co-stimulatory molecules with their ligands on antigen presenting cell is mandatory for T cell activation. Activation of T cell, CD4 T cell, CD4 TCR receptor, CD28 class 2 MHC molecule, signal 1 and signal 2 target cell, B7 family molecule. The next, B cell become anergic. CD4 T cell TCR receptor, CD4 class 2 MHC molecule, signal 1 alone target cell, and signal 2 alone target cell, CD4 B7 family molecule, CD28 CD4 T cell. No effect on T cell. Antigenic peptides presented on MHC molecule signal 1 and co-stimulatory signal 2. Interaction of CD28 on T cells and B7 family molecule on target cells are required for full T cell activation. And there is no co-stimulatory interaction. T cells become non-responsive or anergic. So only signal 2 alone are not sufficient to force T cell activation. Once activated, new T cell undergo clonal expansion followed by its differentiation into effector and memory cells. Then soluble factors influence T cell activation. T cell activation is also influenced by the activity of soluble cytokines secreted by T cell and APCs. The assisting help provided by cytokines, sometimes referred to as signal 3, for T cell activation. The cytokines bind to cytokine receptors and stimulate a cascade of downstream events that enhance T cell proliferation and survival. For example, IL-2 It is a well-known cytokine involved in T cell activation, especially for cytotoxic T cells, CD8 plus T cells. Then T cell differentiation. Once T cells gets activated in response to signal 1 and signal 2, naive T cells will enlarge into a blast cell and begin undergoing repeated rounds of cell division. It divides rapidly for 4 to 5 days, generating a clone of cells that differentiate into effector and memory cells. The naive T cells, effector B cells, and memory cells vary in their phenotype and action as they display broad differences in expression of surface proteins. The function of T cells is majorly done by effector cells, with which help in clearance of pathogen, while memory cells remain quiescent and long-lived but easily activated as compared to naive cells. They respond with a very fast rate to the subsequent challenge, and same antigen are responsible for secondary response. Effector cell lifespan short-lived, a few days to few weeks. Memory cells long-lived, months to years. Protection against ongoing protection. Primary response. Effector cells of CD4 plus T cells secrete cytokines that help several other cell types such as B cells, macrophages, and CD8 plus T cells. Effector cells of CD8 plus T cells bind and kill infected cells. Then memory cells against a secondary infection, secondary response. Memory T cells get rapidly activated in response to subsequent exposure of antigen and results in faster T cell response, secondary response. Then sites of location, secondary lymphoid organs, and sites of infection, secondary and tertiary lymphoid organs. Types of effector cells and their function. T helper effector cells. T helper effector cells. The naive CD4 plus T cells get activated in response to following signals. Antigen specific signal received by TCR complex upon recognition of specific peptide present on class 2 MHC molecules on a professional antigen presenting cells. Echo stimulatory signal committed by the CD28B7 family members as described above. A cytokine signal mostly secreted by activated APCs neighboring cells at the site of location. Activated T cells which binds to cytokine receptor, which results in proliferation and differentiation of the antigen activated CD4 plus T cells into distinct CD4 plus T effector cells. The CD4 plus T effector cells predominantly acquire the ability to secrete factors that enhance and modulate the activation and proliferation of other cells. These CD4 plus T cell effector cells are also known as helper T cells. The CD4 plus T effector helper cells are very heterogeneous. The heterogeneity mainly occurs due to signal 3, cytokine signal. The T cell activation is highly influenced by additional factors like cytokines secreted by APCs or neighborhood cells. Which cytokine is released by APCs depend upon type of APC, dendritic cell, B cell, or macrophage. Surrounding neighboring cells like mast cells, natural killer cells, type of infection, site of infection, etc. The different cytokines activate different downstream signaling pathways, which in turn influence expression of different sets of genes, resulting in distinct effector helper cells. The effector helper cells are very heterogeneous in phenotype and function. In fact, based on distinct sets of factors secreted by T helper cells, these are well characterized distinct functional subgroups Th1 and Th2. The cytokines released by one subset inhibit the development and activity of opposite subset. The phenomenon is known as cross-regulation. For example, IFN gamma released by Th1 inhibits proliferation of Th2 subset. Similarly, IL4 released by Th2 downregulates secretion of IL12, which requires for Th1 subset differentiation. Additional T helper cell subsets are also developed in the presence of specific sets of cytokines, which influences different sets of genes in activated naive T cell and help to differentiate in different subsets. For instance, Th17 cells are generated when naive T cells are activated in the presence of IL6, IL23, and TGF-beta, while TGF-beta along with IL2 results in formation of induced regulatory cell, induced Th-reg. Both subsets have opposite function, where Th17 cells produce IL-17A, IL-17 and IL-22 cytokines associated with chronic inflammatory and autoimmune response such as arthritis, multiple sclerosis, while induced Th-reg secretes IL-10 and TGF-beta, which downregulate inflammation by inhibiting activated T cells and APCs. Similar to Th1 and Th2 subsets, Th-reg and Th17 can also cross-regulate each other. Th17 contributes to inflammation and autoimmunity. Induced Th-reg inhibits inflammation. Then Th17 protects against fungal and bacterial infections. Th-reg protects against chronic inflammation and autoimmune response. Follicular helper T, TFSH cells. FH cells are the new addition to T helper cell subset. TFSH cells are generated from activated naive T cells in the presence of cytokines IL-6 and IL-21. Like TTH2, TFH cells play important role in B cell health and are found in B cell follicles and germinal centers. The effector cytokines released by TFH cells are IL-4 and IL-21. So activated naive T helper cell differentiate into different subset of effector helper T cells in the presence of certain cytokine. Differentiated subset of T helper cells further secrete characteristics cytokine which has wide range of effects. Cytotoxic T effector cells. CTL cytotoxic T lymphocyte. The naive CD8 plus T cells get activated in response to following signals. Antigen specific signal received by TCR complex upon recognition of specific peptide presented on class 1 MHC molecule on a target cells. The co-stimulatory signals transmitted by the CD28B7 family members as described. So at cytokine signal, IL-2 mostly secreted by activated T helper cells which binds to IL-2 receptor which results in proliferation and differentiation of the antigen activated CD plus T cells into CD8 plus T cells, effector cells and memory cells. Target cell class 1 MHC molecule, B7 family molecule, CD8, TCR receptor, CD28, CD8 plus T cells, activated CD4 plus T cells, then activated CD8 plus T cells, differentiation effector CD8 plus T cells, memory CD8 plus T cells. The CD8 plus T cells gets activated in response to antigen presented by class 1 MHC molecule. Co-stimulatory interaction between CD8 plus T cells and target cells and IL-2 cytokine secreted by activated CD8 4 plus T cells. The CD8 plus T effector cells predominantly acquire the ability to kill the target cells expressing the processed antigen on class 1 MHC molecule. The CD8 plus T effector cells are also known as killer T cells or cytotoxic T lymphocytes, CTLs. Mechanism of CTLs killing. CTL effector cell first recognizes specific peptide MHC complex on target cell via TCR complex and forms a cell-to-cell conjugate. This conjugate event is followed by the induction of apoptotic death in target cells. After that, CTL effector cell dissociates from the dying target cell and goes on to bind another target cell. There are two major ways by which effector killer cells induce apoptosis in target cells: granzyme and perforin-mediated cell killing. After conjugate formation, mostly CTLs start secreting cytoplasmic granules rich in pore-forming protein perforin and several serine proteases called granzymes into the space at the junction between two cells. The perforin molecules contact the target cell membrane and makes holes in the membrane. The granzyme molecule make entry in the cytoplasm via the perforin-made holes or via endocytosis process. The granzyme molecule initiate the events that activate apoptotic pathway within the target cell. Target cell begin to exhibit DNA fragmentation. Cytotoxic T cell, granzyme, perforin. Target cell activates apoptosis pathway. So granzyme and perforin mediate killing mechanism by cytotoxic T cell. Cytotoxic T cell releases cytoplasmic granules rich in pore-forming protein perforin and several serine proteases called granzymes into the space at the junction between two cells. The perforin molecule contact the target cell membrane and makes holes in the membrane. The granzyme molecule makes entry in the cytoplasm via the perforin made holes. The granzyme molecule initiate the events that activate apoptotic pathway within the target cell. Then Fas-ligand mediated killing. After conjugate formation, some CTL mediate killing via the expression of Fas-ligand on their surface. The Fas-ligand belongs to tumor necrosis factor family. The Fas-L interact with the Fas-CD4-35 on the surface of target cells, which delivers a death signal in target cell. The interaction of Fas with Fas-ligand leads to the association of Fas with adaptor molecule FAD. So, Fas-associated protein is the death domain, which in turn result in activation of caspase cascade leading to apoptosis of target cells. Cytotoxic T cell, Fas ligand, Fas or Fas receptor. CD45, target cell activation of caspases, apoptosis, FAD. Fas ligand mediated killing mechanism by cytotoxic T cell. Here, cytotoxic T cell mediated killing via the expression of Fas ligand on their surface. The Fas interacts with the Fas CD95 on the surface of target cells, which delivers a death signal in target cell. The interaction of Fas with Fas ligand leads to the association of Fas with adapter molecule FAD-D, Fas associated protein with the death domain, which in turn results in activation of caspase cascade, leading to apoptosis of target cells. In summary, T cell activation is referred to changes that occur in T lymphocyte following antigen recognition. The naive T cell recognize antigen on MHC molecules via T cell receptor TCR complex along with the help by additional molecules such as co-receptor CD4 in case of T helper cells or CD8 in case of cytotoxic T cells and adhesion molecules. It is followed by downstream signaling cascade. Various signal transduction pathways involve Fas protein followed by MAP signaling pathways, protein kinase C followed by NK beta pathways, IP3 mediated pathways, etc. Apart from binding of TCR and CD8 CD4 molecule to peptide MHC complex, additional co-stimulatory signal is required for T cell activation. Without co-stimulatory interaction, T cell become anergic. Together, presence of antigen along with co-stimulatory interaction leads to cell proliferation, cytokine secretion, and differentiation of T cell into effector or memory cells. T cell activation is also influenced by the activity of soluble cytokines secreted by activated APC, neighboring cells at the site of location activated T cells. The assisting help provided by cytokines, sometimes referred to as signal 3 4 T cell activation. The CD4 plus T effector cells predominantly acquire the ability to secrete factors that enhance and modulate the activation and proliferation of other cells. Thus, CD4 plus T effector cells are also known as helper T cells. These effector helper T cells are very heterogeneous in phenotype and function. In fact, based on distinct sets of factors secreted by T helper cells, there are well characterized distinct functional subgroups such as Th1, Th2, and I Treg cells. The CD48 plus T cells get activated in response to antigen presented by class 1 MHC molecule. Co-stimulatory interaction between CD8 plus T cells and target cells and IL-2 cytokines secreted by activated CD4 plus T cells. The CD8 plus T effector cells predominantly acquire the ability to kill target cells expressing the processed antigen on class 1 MHC molecule. The CD8 plus T effector cells are also known as killer T cells or cytotoxic T lymphocytes. The cytotoxic T cells secrete pore-forming protein perforin and granzyme or expresses Fas ligand to activate apoptosis in target cells.